Okay, so now we're moving on to Chapter 25, Population and Evolutionary Genetics. So first let's do an overview of what population and evolutionary genetics refers to. So you guys probably remember from general biology, you've learned about evolution, and you know it's a consequence of changes in the genetic material through mutation and then changes in allele frequencies in populations over time. Now, most everybody, when they think of evolution, the first thing they think about is natural selection. And natural selection has a huge impact on evolution, but there are other forces, including migration, mutation, and genetic drift. And these all three, individually and collectively, alter these allele frequencies and bring about evolutionary divergence that eventually leads to the formation of new species called speciation. And we're going to talk about all these different things, you know, later in the chapter. There's two main fields of evolution. You have microevolution, which is changes that are occurring within a population or a species. Then macroevolution is larger evolutionary events that lead to the emergence of new species. So let's first talk a little bit about genetic variation in populations. So a population is just a group of individuals that belong to the same species that live in a defined geographic area and mate. All the gametes that are being produced by all these people in the population constitutes the population's gene pool. So all those alleles, all that genetic information that's being carried by individuals constitutes the gene pool. And most populations contain a very high degree of heterozygosity. And so, you know, even though it may not be seen on a phenotypic level, we're all carriers of these alleles that you don't see the phenotype for because they're recessive. So one of the ways that you can see how much genetic variation is present in a population is to use artificial selection. So if the population is a lot of homozygotes, meaning they're not carriers of these recessive alleles, they're not heterozygous for these different traits, and you try to use artificial selection to tease these traits out, then it's not going to work, and the phenotype's really not going to change. But if you have a lot of heterozygotes carrying these recessive alleles and you don't see them in the phenotype, but you do some breeding studies and do some artificial selection, you'll see that the phenotype can change very quickly over just a few generations. And the best example is domestic dogs. And so we know that this tiny little Chihuahua and this great big Great Dane, both arose from wolves, and not that long ago, evolutionarily speaking. And so while they look completely different, there's a lot of variation there. They really came from the same thing. So those wolves had a lot of genetic variation kind of hidden in their genome. Now, also what you don't see on the phenotypic level is the variations that are present in nucleotide sequences. And so we can look at the actual nucleotide sequences, the ATCG, of genes carried by individuals in a population and see how much variation is there. And it should be a lot more than just what you see in the phenotype. A good example is alcohol dehydrogenase in Drosophila. So this is just a gene in which an enzyme where there's only really two alleles present in Drosophila. There's the ADHF and the ADHS. And the two enzymes that are produced by these two different alleles only differ by one single amino acid. So some researchers wanted to look on the nucleotide level and see if that difference in phenotype, that difference in proteins of just one amino acid, corresponded to the variation at the nucleotide level, at the sequence of bases. So they took they cloned and sequenced genes from Drosophila of 5 different populations, and they found a total of 43 nucleotide variations that were distributed throughout this single gene, alcohol dehydrogenase. Now, a majority of these were found in the non-coding regions. So we're talking about these introns and the untranslated regions on the ends. So a majority of them were not in any kind of region that would code for a protein. But there were 14 nucleotide substitutions that they found within the coding regions, within the exons. But remember, there's only 2 alleles, and they only differ by a single amino acid. So it turns out that 13 of the 14 nucleotide substitutions resulted in a change in bases, but did not code for a different amino acid, because remember, the genetic code is degenerate, so you can have multiple codons that code for the same amino acid. So these mutations are considered silent mutations. And so we can see here you have the 5 different Drosophila populations. Now, the little dots represent where the amino acid, or not the amino acid, excuse me, the DNA sequence was the same as what's present in the consensus sequence. But there's one here. This one here is the only one that actually resulted in a phenotypic change, a change in amino acid sequence. So out of the 13 of these mutations that were changed the nucleotide sequence, it did not change the amino acid sequence except for in this one instance. Another good example is cystic fibrosis in humans. So the cystic fibrosis is caused by mutations that occur in the cystic fibrosis transmembrane conductance regulator, which this is a protein that's a transmembrane protein that actually serves as a chloride ion channel. And if the protein is non-functional and the chloride ions aren't moved in and out of the cell the way they're supposed to, it leads to cystic fibrosis. And so this is a recessive loss of function mutation in the gene that causes cystic fibrosis. And of course, you have to have 2 alleles with these mutations in order for you to have the disease. They found that actually there's more than 1,900 different mutations in this gene, and they're all different kinds of mutations. So you have missense, deletions, nonsense, frame shifts, splice defects. All these things are present. Now, a majority of people that have cystic fibrosis, it comes from a particular mutation called delta F508, which is present in exon 10, and it's a 3 base pair deletion. That accounts for 67% of the alleles in the population that lead to cystic fibrosis, but there's a lot of other ones out there as well. The take-home message here is that there's a lot more nucleotide variation that exists than what you see at the phenotypic level. And this is just a diagram that's in your book that shows each line here is a different mutation, and down here at the bottom it shows you like what region of the protein, because the protein has a lot of functions. You know, it inserts itself in the membrane, so there's certain regions that are membrane spanning. ATP has to bind to it in order to it's an active transporter, so it requires energy, so it has to bind ATP. Here's another membrane spanning region here. You know, so you've got all these different regions to the protein, and so then these are all the mutations here that, you know, are present in these different regions. And you can see here, this mutation is the one that's the most prevalent. This is the number of, the y-axis is the number of copies. And you can see that the delta F508 is the most prevalent allele. So how can you have all this genetic variation that you don't really see? Well, this is the neutral theory of molecular evolution. It was proposed by Moto Kimura in 1968. And what he basically said was mutations that lead to these changes in the amino acid sequence are usually detrimental. Like, for example, the cystic fibrosis ones are detrimental. And there's only a few small fraction that are favorable. The majority of these mutations are neutral, meaning they're functionally equivalent. They don't really affect the organism in a positive or negative way. The ones that do create a positive or negative effect are going to either be preserved or removed from the population by natural selection. But the neutral alleles are going to be there based on, you know, the mutation rates and something called random genetic drift, which we're going to talk about later. This is just by chance. So the diversity that you find at most loci doesn't always reflect the action of natural selection. Instead, it's a function of the actual population size and the fraction of mutations that are neutral. Now, some of these mutations that we consider neutral turn out to be almost like savings accounts for the individual. What looks like it's not important later on in a particular environment may actually save that individual. For example, in the next lecture we're going to talk about mutation in a receptor on a cell that makes people resistant to HIV. Now, normally under an environment where HIV didn't exist, that mutation wouldn't be favorable at all. And in fact, it would probably be fairly neutral. But in the instance of an HIV epidemic, that becomes a favorable allele that gets chosen. So all these genetic variations that don't seem to matter under certain environmental conditions may actually become very important. So it's almost like Mother Nature's savings account. Okay, so that's it for this lecture. We're going to answer a few little questions and then move on to talking about the Hardy-Weinberg law.